Good afternoon, respected director, sir, faculty members, dear students and audience. I, Dr. Sant Kumar Verma, Associate Professor, ISF College of Pharmacy, would like to welcome Dr. Rohit Seth, Associate Professor, School of Life Sciences, Guru Ghasidas Central University, Bilaspur, at the online platform of ISF College of Pharmacy for delivering talk on neurobiology and obesity and leptin resistance. Sir has more than 20 years teaching and research experience in the area of cell and cancer biology, neuroscience, and data analysis. Sir, I welcome to you on ISFCP online platform. Over to Dr. Rohit, sir. Sir, please.
Okay. Thank you, Dr. Sant Verma, for inviting me for this lecture. As already shared my topic, I will be delivering a talk on neurobiology of obesity, and we'll go from there. Let's start the presentation. Let me share this presentation. Is my screen visible to you all?
Yes, sir. Yes, sir.
Okay. So the topic is neural control of obesity and leptin resistance. So, as a student of pharmacy and biology, most of you must be aware of what leptin resistance is. If not, definitely at the onset of this presentation, you'll get to know about it in a while. So, let me start with my first slide. The question starts, why do we eat? And many of the people, if you ask this question, they will tell, I eat because I'm hungry, right? I think that's a common assumption. People will say, I'm hungry, that's why I'm eating. Now, the next question comes immediately, why are you hungry? Another answer, I'm hungry because I have not eaten. Come on, that's not the thing. I wanted to know the exact logic behind it. Why do we eat and why I'm hungry? So, why we eat? Because of certain physiological needs and also certain psychological factors. Physiological needs are need for metabolic fuels as an energy source, need for amino acids, for protein turnover, for vitamins and minerals to replace the losses, and psychological factors are appetite, hunger, satiety, and some social functions. For example, you go to some social function and you are tempted by some food. So that is another thing that can add to the psychological factor. Hedonic value of the food for the pleasure. For example, you are going out and just for pleasure you are having some food that you are not taking on a routine basis. And then psychological value of the food. Okay, sometimes you don't eat, but under peer pressure also you eat certain things. So the purpose of getting this food is to get some kind of energy. And the main energy sources from our food are from carbohydrates, proteins, fats, alcohols. And depending on the type of sources, they provide different kinds of energies. For example, with carbohydrate, we get 4 kilocalories per gram of energy. Similarly, by fat, we get 9 kilocalories per gram of energy. Okay, now this slide is showing a section of the brain, a mammalian brain, in which the main area which is pointed by this pointer is reflecting the hypothalamus. This is the hypothalamus part of the brain. And this is a very crucial part of the brain that is involved in the regulation of food, eating, and also satiation. Like this, the red dots on the sides, it is called lateral hypothalamus, or it is also known as hunger center. That means whenever this center is active, the person will feel hungry. And similarly, there's another center right below this. It's known as ventromedial hypothalamus. It's also called as satiety center. Okay, so that means whenever I'm full, this center becomes active and it will say stop eating. So this is the satiety center. So the short-term signals that are coming from or activating on these centers are direct nerve inputs that are coming from the GI tract and the liver. So they are also producing certain factors that act on these centers and thereby regulating the food intake. There is also certain plasma concentration of certain metabolic fuels that is also governing the activity of hunger and satiety center. And then relative concentration of insulin and glucagon. We all know about these two hormones. They are produced by the pancreas and they are regulating the level of glucose. So concentration of insulin and glucagon in the blood is also regulating the activity of hunger center and satiety center. Then there are certain peptide hormones. These are secreted by the gastrointestinal tract during the digestion process. Some hormones are like ghrelin, then pancreatic peptide YY, then we have cholecystokinin, then so many different hormones. We'll talk in brief about it. Okay, so in this slide, this is a very important slide in which you are seeing two mouse. There are two mouse. One mouse is extremely obese, the other mouse is lean. Okay, so this is a obese mouse and this is a lean mouse. Okay, so scientists found that from the same cohort of animals, how come one individual becomes so much obese, the other individual remains lean? So later on, they found out by the study from Zhang et al. In 1994. They cloned one gene, it is called OB gene. Okay, so this mouse was found that there is the OB gene is mutated. The OB gene is mutated in this mouse, and later on, it was found due to lack of the product of OB gene, this animal becomes extremely obese. And Zhang et al. Also cloned this gene and found that it is secreting a peptide that has got a signal sequences. It has got a signal sequence that means it is secreted, some peptide that is getting secreted. So injection of this peptide into the obese mice started him to lose the weight. So because this obese mouse started losing weight, this is called leptin. The name was given leptin, which in Greek means lean. So initial studies showed that it acted on hypothalamic appetite centers, thereby is depressing the appetite and signaling the state of adipose tissue reserve. Now, interestingly, this leptin is secreted from the obese tissue, that is adipose tissue, right? So it is coming out from the adipose tissue. So it is also telling about the reserve of adipose tissue in the body. So it also regulates the energy expenditure by uncoupling the oxidation from ATP formation. So the great excitement was that the leptin or leptin agonist would be a cure for obesity. But the glitch is here that the obese people, they secrete higher than normal amount of leptin because obviously they have a lot of more adipose tissue. Okay, so the scientists, when they heard about it, they became very excited and they found that this is a fascinating thing. Anyone who is obese, you just inject leptin in their body and they will start losing weight. But the problem lies here. Try to understand this thing, which is a very simple logic. If this leptin is coming from adipose tissue, that means the obese individual must be having a lot of leptin. I think I make a sense because there is obviously a lot of adipose tissue that is present And if there is a lot of adipose tissue that is present, that means there is a lot of leptin that is present already in the body of an obese individual. Then how come this leptin is not able to bind? That means there is something else going on. So they later on, the scientists found another category of animal which is known as a DB or a diabetic obese mouse. DB stands for the receptor for leptin. So either leptin is absent, or if leptin receptor is absent, in both the cases the individual becomes morbidly obese. So, how do you know about these individuals? They become obese because they become hyperphagic. That means they're always hungry. They always like to eat more food. Okay, and these individuals, they also suffer from type 2 diabetes. That means they are not only hyperleptinemic, they are also hyperinsulinemic and hyperglycemic. So, an experiment was performed in which the scientists took one lean animal and one obese animal, and they performed an experiment called parabiosis, in which they cut the body wall of both the individuals and stitched it together side by side. So when they stitch them together side by side, what happens? Their blood circulation also starts to become to exchange. So once the blood circulation starts to exchange, then the amount of leptin or leptin receptor, that is also getting exchanged in between the individuals. So like this, there was no effect on the lean animal, but the diabetic individual that lacked the leptin receptor, or a OB individual that lacked leptin, they started to lose weight. That means there is some factor that is coming out from the body of a lean animal and going into the obese animal, and they start to lose weight. Okay, so we already discussed about this hunger center and the satiety center. This hunger center is primarily controlled by one set of neuropeptide known as neuropeptide Y, which is coming from the hunger center. This is responsible for increasing the food intake and reducing the energy expenditure. Contrary to this, there's another group of neuron which is known as melanocortin neuron, which is coming from the satiety center. This decreases the food intake and increases the energy expenditure. Okay, so leptin, what does it do? The leptin is going to inhibit the expression of neuropeptide Y. So if it inhibits the expression of neuropeptide Y, that means the person is not becoming too much hungry, right? Rather, it is activating the melanocortin neuron. That means it is activating the satiety center. Okay, now it's time to define the obesity. So any malfunction in the energy balance and the energy expenditure can be called as obesity. Okay, and why it is important? Because obesity is directly linked with so many different problems like cancer, gallbladder disease, renal failure, stroke, heart failure, and so many non-insulin dependent diabetes mellitus. All these problems are directly linked with obesity. So, how does the central nervous system control food intake and cardiovascular functions? So, this is the question that we asked in our research, right? So, we have just now seen a hunger center and a satiety center. What are the molecules that are working on the hunger center and the satiety center? Now, we will look a little bit more closer. What are the molecular mechanisms that are involved in regulation of food intake? For example, if we see, this is a CNS mechanism that is linked with the energy balance and cardiovascular function. If we are having a reduced caloric intake, that means food intake is less, then this is directly linked with the decreased energy expenditure, decreased sympathetic activity, decreased heart rate, blood pressure, and also obesity and cardiovascular disease. Contrary to this, if there is overfeeding, we are having a greater amount of energy balance, increased sympathetic activity, the heart rate goes up, blood pressure goes up, and also obesity as well as cardiovascular disease risk is increased. So that means, what is energy balance? Energy balance means simply, for example, you are taking say 200 kilocalories of energy in terms of food, and you are utilizing only 120 kilocalories of energy. So remaining 80 kilocalories of energy which is left over, it is stored as fat in form of energy balance. Okay, let's see certain key players that are involved in regulation of this food intake. So first we have seen the leptin. It was discovered in 1994. It's a 16 kilodalton protein hormone that plays a key role in regulating the energy intake and energy expenditure, including appetite and metabolism. Leptin is secreted from adipocytes and acts as a long-term adiposity signal. And absence of leptin in mice and human as a result of genetic mutation produces a obese phenotype. So if we see this from hypothalamus, we are seeing that this is leptin is produced from the adipose tissue. Whenever there is an increase in fat, there is an increase in the expression or production of leptin. This leptin goes and binds in the hypothalamus. Thereby, it increases thermogenesis and decreases the food intake. Okay, so as a result of which, there is less fat deposition in the body. And what are the areas I just mentioned? In the hunger center, we have this hormone, this group of nerves known as NPY and AGRP producing neurons. And in the satiety center, we have POMC, pre-opiomelanocortin and CART, cocaine and amphetamine regulated transcripts. So they are directly connected with the lateral hypothalamic area. This is directly connected with the paraventricular nucleus. Okay, so the leptin inhibits the NPY and stimulates the POMC neurons, thereby inhibiting the food intake. So in general, the obese people have unusually high amount of circulating concentration of leptin. These people are said to be resistant to the effect of leptin in much the same way that the people with type 2 diabetes are resistant to the effect of insulin. The pathway of leptin control in obese people might be flawed at some point so that the body doesn't adequately receive the satiety feeling subsequent to eating, and this is known as leptin resistance. So we have already known now in a nutshell, if a person is obese, that means its body is having a lot of adipose tissue and also a lot of leptin. Okay, so what does this leptin resistance mean? How does it happen? There are so many theories regarding that. Like there will be circulating regulators, like there are circulating leptin receptors, and their level of these receptor goes down. There is upregulation of certain molecules like SOX3 and PTP1B. They are also actually contributing to leptin resistance. Then leptin induced leptin receptor down regulation, that is another thing. And also we have just now discussed mutation like OB and DB mutations. So either there is a leptin mutation or there is a leptin receptor mutation. All of this contribute to leptin resistance and hyperleptinemia. For example, I gave you just example of SOX3. Okay, so SOX3, if you see, this is when there is a low level of circulating leptin, then there is a proper signal transduction pathway, which is JAK-STAT pathway, that is also regulating the PI3 kinase pathway, and appropriate phosphorylation of this tyrosine residue is seen. Now, whenever the level of leptin goes up, this STAT3 at this tyrosine 1138 position, this is contributing to the activity of SOX3 or elevating the activity of SOX3, which in turn can down-regulate the actual signal transduction mechanism, down-regulating the JAK-STAT pathway and also PI3 kinase pathway. So whenever there is a chronically high level of circulating leptin, the SOX3 level goes up, and also down regulates the actual signal transduction mechanism. Okay. So the other satiety signals, they include leptin we have just now discussed, that is directly acting on AGRP, NPY, and POMC CAR-T neurons. And then apart from that, there are other molecules also like PYY, GLP-1, oxyntomodulin, pancreatic peptide, amylin, ghrelin, so many different hormones coming from the gut that are also regulating the food intake. So the main question to be asked here was, we have already seen that there is a leptin resistance. Now, in most of the obese individual, that's the case. Now, how we can get rid of this leptin resistance? That is the main question. So while I was working as a postdoc, there a new fascinating molecule came up in the light that was known as amylin, and amylin is also coming from the same pancreatic cells. So they are also secreted from the beta cells. It's a 37 amino acid peptide that has got a glucose regulatory and anorexigenic action. That means if a level of amylin goes up into the blood, that means the person is going to eat less. It is having anorexigenic action. So it acts as a short-term or episodic satiety signal. So amylin is produced from the pancreas, and whenever it is coming into the circulation, it goes and binds into the hindbrain area. Okay, we just saw the leptin is binding in the midbrain area in the hypothalamus. Amylin is binding in the hindbrain area, in the area postrema and the nucleus of solitary tract. So what does it do? It is going to decrease the food intake. It decreases the food intake. Thereby, it's also increasing the activity of glucagon and insulin. So thereby maintaining the level of glucose into the blood. So what amylin will do? It will down-regulate the level of glucagons. So we have done this study in obese rodents, in which one group of animal was lean animal, in which amylin was introduced. And whenever the amylin was given, you can see from day one itself, there is a steep decrease in the food intake. And this decrease was almost maintained for 24 days. So the triangles, the open triangles here, they are the control groups, and the closed circles here, they are the amylin group. So this is the story in the lean animal. Now coming to the interesting part, which is the obese animal. We have already seen that the obese individual, which are leptin resistance, they don't lose the weight. But when they are provided amylin, immediately the level of food intake goes down, and this lower level of food intake is maintained throughout the study for remaining 24 days. Okay, so once the leptin resistance has developed, what can be done the next? Treatment. So what is the treatment? Let's coming to the treatment part. Now, many scientists, when I was working on this area, many scientists around the world were working on different types of combination therapies. So non-clinical and clinical studies have already provided the evidence that leptin plays an important role in body weight regulation. But for obesity management, when the leptin was given to the obese individual, it was proven unsuccessful. So the combination therapy came, and a translational research program aimed at elucidating the interaction between different islet, gut, and adipose-derived hormones led to the discovery that combined amylin and leptin agonism induces the marked synergistic, fat-specific weight loss in leptin-resistant human and rodents. And this study proves the same in the human. So pramlintide stands for the amylin, and metleptin is the recombinant leptin. So whenever you can see leptin is given, this is the first one, there is a modest decrease in the body weight. But when pramlintide or amylin is given, there is a decrease, This is not in the obese, by the way. It is in the lean and individuals, in the lean individuals. But with these lean individuals were provided pramlintide along with metleptin. You can see the closed squares in the bottom. There is a dramatic decrease in the body weight of the individual, which is a synergistic effect, you can say, of amylin and leptin together. So combination therapy seems to be effective. So, on the basis of this study, we designed our own study in animal with an objective to study the effect of leptin or amylin or combination of leptin and amylin on food intake, body weight, metabolic and cardiovascular parameters in lean and obese FB and F1 rash. So these Fisher Brown Norway rats, they are very specific rat. They don't show. They showed modest obesity. Doesn't show like really morbid obesity, just like Sprague Dolly rats. So for that reason, this animal was chosen. So the lean animals were fed on 4% fat standing chow. Then obese animals were fed on 32% fat diet. They were housed in metabolic chambers. I'll show you the metabolic chambers. So this is the scheme of the experiment where surgery was performed. Surgery means there is a implant of telemetry device inside the body that tells us about 24/7 their heart rate and then change in the respiratory quotient. So surgery was performed, 7 days they have left for acclimatization. Then animal were placed in the metabolic chambers. Baseline data was collected, and then injection was provided for 7 days, amylin and leptin injection. And after the end of 7 days, the animals were sacrificed. So you can see this is the telemetry device that was inserted inside the body of the animal. It's connected with the dorsal aorta, and these are the metabolic chambers in which the animals were placed. This is a close look of the chamber where all the facilities provided. Each sip of water, each bowl, each amount of food is calculated. You can see this is sealed properly for the indirect calorimetry. The locomotor activity of the animal is also recorded on this platform. There is a thermostat that regulates the temperature inside the cage. There's a fat. And then the drug was provided with the help of osmotic pump for sustained release. Okay. So there is a sustained release. Once you fill the drug, place the osmotic pump inside the body of animal. Gradually, the drug is getting released for the remaining 7 days. So this is the result from the lean animal. You can see these are the open circles. These are vehicle. Then closed triangles is the treatment, and open triangle is the pair fed. Pair fed means that group is they are going to pair fed to this particular group. The amount of food that is consumed by this group is the same amount of food that is provided to the pair fed group. So you can clearly see with the leptin there is a modest decrease in the food body weight. There is a little more decrease in the body weight with the amylin, and dramatic decrease with the in the body weight with the both leptin and amylin combination. Then coming to the caloric intake, you can clearly see from this data that there is a drop in the caloric intake that's maintained for rest of the experiment, and combination therapy again appears to be highly efficient in maintaining the caloric intake. Now coming to the obese individual, the modest decrease in the in this group was seen in leptin, but when in seeing an obese individual, there is no change in the body weight or in the caloric intake of the leptin injected animals. Rather, there is a decrease in the body weight and also food intake in the amylin treated group, which is highly significant in the combination group. Both body weight as well as caloric intake is down in the obese individual, or in other words, you can say in leptin resistant animals, the combination therapy is working miraculously, decreasing the body weight as well as food intake. So we reported that the combination of leptin and amylin exerted a greater anorexigenic effect and weight reducing effect than either peptide alone. So, in lean and obese rats, subcutaneous administration of leptin amylin reduces respiratory quotient, which is very important parameter to be taken into account, representing that there is a fat metabolism, lipolysis. So, whatever amount of food in the body weight is going down, it is because of burning of the fat, okay, not the proteins or the lean mass. So this was also represented in the form of fat mass from lean and obese individual that was taken out at the time of sacrifice. You can clearly see these black bars are representing amylin and leptin combination both in lean and animal obese individual. You can see mesenteric, retroperitoneal, epididymal, as well as total visceral fat is going down with the treatment of amylin and leptin. So this is also, you can see the distribution of fat. This is retroperitoneal fat, the epididymal fat. Okay, in the saline treated or the control group, this is amylin and leptin treated group, where barely you can see any fat left in the body. And pair fed individuals which are provided same amount of food but no drug, they are not showing any change in the fat pad weights or amount of fat pad present, right? So that means the amount of dissolution that is coming here, or the lipolysis, is due to the presence of amylin and leptin. Okay, I will quickly skip this study. We have also performed the cardiovascular study, but we will just focus on the obesity part. So this work is already published in International Journal of Obesity. Now the question comes here, how amylin is able to restore the leptin responsiveness. Okay, because we already mentioned that there are two different binding sites. No, the amylin is binding in the hindbrain, leptin is binding in the midbrain. But what is the mechanism? How does it start to restore the leptin responsiveness? So we came to this hypothetical model where we are saying that amylin goes and binds to its receptor in the area postrema nucleus of solitary tract, thereby activating the formation of CGMP and cyclic AMP. Thereby, some signals are transmitted from hindbrain to the midbrain, or there is some kind of crosstalk in between this amylin CGRP receptors and histamine receptors. Okay, so leptin is binding in the midbrain, and amylin is actually activating this particular circuit in order to make this functional, and thereby increasing the food intake, lipolysis, and energy expenditure. So there are several instances. There are so many papers that were coming at that time when we were working, that it is saying that it is the histaminergic signals that are actually regulating the food intake in the animals. So thereby we also focused on some histamine activity. And in this slide, you can clearly see that this is the normal saline or a controlled individual. This is the arcuate nucleus, and this is the ventromedial hypothalamus. Here, you don't see any activity of histamine or a bare minimal activity of histamine. But when you are actually injecting this amylin plus leptin treatment, you can see a lot of positive cells for histamine in both arcuate nucleus and in the ventromedial hypothalamus. Similarly, the immunohistochemical localization of histidine decarboxylase, the enzyme that is actually regulating the production of histamine, was also going up in the arcuate and the ventromedial hypothalamus, uh, and also in the nucleus of solitary tract. You can see all these positive cells in area postrema, in the ventromedial hypothalamus, in the arcuate nucleus after 1 day infusion of amylin and leptin in the animals. Uh, this is showing you the histidine decarboxylase and CFOS activity, early expressions in the tuberomammillary nucleus. Tuberomammillary nucleus is the site where histamine production actually occurs. So it is clearly showing you this these cells are fully loaded with the histamine, and also the CFOS activity is really high, or histidine decarboxylase activity is also correspondingly high in this particular cells. So this is the control group. And the bottom you are seeing the APL group. This is the control or saline treated group. Very few cells are there. Here the loaded cells are there, APL group or amylin plus leptin, and here's correspondingly CFOS activity is also high in the same cells. Similarly, here you are seeing in the paraventricular nucleus, in the saline treated animals, in the amylin plus leptin treated animals, the amount of the HDC or histidine decarboxylase positive cells is high in the this APL group or amylin plus leptin group. Now we wanted to see in real time what is happening in the brain. So we actually injected the animals with amylin and leptin, and we wanted to see what is happening in the real time in the brain of these animals. For this, we have inserted a cannula, placed a cannula inside the brain, targeting the ventromedial hypothalamus, and we injected the cerebrospinal fluid into the brain, which washed out this particular area, and we collected the sample with by the procedures known as microdialysis. So in this experiment, you can clearly see that the cannula is inserted into the brain of animals, and with the micro injector, we inject the cerebrospinal fluid with the help of these linings. And there is a small tubing which is coming out from this setup, and it is collecting all the wash out from inside the brain in the ventromedial hypothalamus, getting collected in these tubes, and immediately these tubes are frozen. So this is how it happens. The CSF is injected, the microdialysis membrane wash out all the CSF stuff in this particular region, and whatever comes out from this region is collected in the form of fractions. Okay, so this is collected here in the form of in the fraction collector. So we try to see what is there in these fractions, and we came to know that in leptin, amylin, and amylin plus leptin, this compound which was detected the maximum, which is known as acetylhistamine. The acetylhistamine was collected. A peak was seen in with the help of HPLC, and we found that this amylin plus leptin showed the acetylhistamine. So this added another component to the study, thereby that there could be an epigenetic factor that is involved in actual regulation of food intake and body weight. So this is brain slice showing the placement of the probe in the ventromedial hypothalamus. Then real-time PCR of whole hypothalamus also suggested that in saline treated group as well as in APL group, you can clearly see the histidine decarboxylase activity and histamine H1 receptor activity. They are going up dramatically. So with this study, we concluded that leptin coming out from adipose tissue and amylin coming from pancreas, they together increase the level of hypothalamic histamine that is binding to histamine H1 receptor. The site of activity is ventromedial hypothalamus and arcuate nucleus, and thereby together they are decreasing food intake, increasing lipolysis, and increasing the energy expenditure. So this work was published in Brain Research. I think this will be my last presentation slide, and this is it. There are some future studies that I'm not going to discuss here at this platform. So if there is any question or query, I can definitely answer that.
Uh, Thanks, sir, for Excellent talk on obesity and leptin resistance. Our pharmacology students and researchers who are working in the area of obesity as well as diabetes also, they definitely get benefited from this lecture. Sir, thanks for sparing your valuable time for ISFCP 101 lecture series. Uh, I would like to request audience, if you have any query related to lecture, please write in the chat box. We will reply after discussion with the expert. So at last, I would like to thank ISFCP management, director sir, and IQSC coordinator, Dr. Siddharth Mehan, our IT team, for their support in conducting this lecture. Thank you, sir, for accepting our invitation and such a nice lecture on leptin resistance and obesity. Thank you, sir.
Thank you so much.
Thank you, sir.